Hi, thanks so much for coming to my talk today. I'm Kati Healey. I am currently a postdoc at Duke University under the supervision of Scott Swartzwelder, and the majority of the focus of our lab is looking at adolescent intermittent ethanol. So we call this AIE. I'm going to use that term a lot during this talk, but we're really interested in using a rodent model to model adolescent binge drinking and see what the effects are, both behaviorally and neurobiologically. And we look a lot in the hippocampus for our neurobiological kind of effects of adolescent intermittent ethanol. And so I'm going to talk a lot about that today and talk about the enduring effects of that on neuroimmune mediators and specifically cell death machinery, neurogenesis, and some neuroimmune markers that we looked at in the hippocampus and their reversal by Donepezil. So first off, why do we focus on adolescence? So adolescence, we kind of know is this big period of change and neuro changes, right? So the onset is from puberty around 12, 13 in the humans, and it actually goes into the mid-20s, especially for males. This can go into the late 20s as well, up to about 25, especially for frontal development. So brain development in adolescence is really important for your function as an adult, and it's a period of rapid changes. So changes really make it very susceptible to insult and can have long-term neurological consequences if you are during adolescence exposed to these kind of insults, right? So during this period, it's really, really susceptible to insult and injury that can remain throughout the lifetime from there on out. We kind of all associate adolescence, and there's a reason why, with increased risk-taking and sensation-seeking. So this period, you're just kind of figuring things out, moving forward, doing a lot of risk-taking, sensation-seeking, kind of working into who you're going to be as an adult, right? And so that can involve a lot of drinking. And adolescent drinking is really prevalent here in the US and actually throughout the world. So 21% of 8th graders, 42% of 10th graders, and 58% of 12th graders report alcohol use in the past year. This has actually gone up during the pandemic. And so COVID-19 and the resulting pandemic and lockdowns and things like that have actually escalated drinking behavior well beyond this even. But binge drinking, which we define as 5 or more drinks for a male or 4 or more drinks for a female in 2 hours, is really prevalent in adolescence. This seems to be the pattern of alcohol use that's most prevalent in adolescence, is this kind of binge drinking behavior, and then they go through these periods of long abstinence, right? So they're not drinking every day, but when they do drink, they drink in this binge manner, which is kind of heavy loading in a very short period of time. So 5% of 8th graders, 16% of 10th graders, and even 24% of 12th graders kind of engage in this kind of binge drinking activity. So an additional study looked at consumption rates in a subset of 12th graders for the past 2 weeks in this high cohort. And what they found is that greater than 10% reported more or 10 or more drinks per week. And then an additional 5% reported 15 drinks or more per week. So in this small subsets that are drinking heavily, they're drinking very heavily. And they're binge drinking, right? They're not spreading this out throughout the week. They're doing this on the weekends with their friends and then having those periods of abstinence, right? So why do we think this is a problem? Well, we know that adolescent drinking really induces these persistent cognitive, behavioral, and neural changes. We see this in the human population. So this alterations are largely in the domains of memory and frontal lobe function. So frontal lobe function is really important for executive function, decision-making, things like that. And these adolescents that have these drinking behaviors and these persistent changes, we see increased risk-taking even beyond this. There's executive function deficits. And so this kind of comes out as having attentional deficits, arithmetic, visuospatial functioning, and working memory impairments in these adolescents that use frequently. And what's even more striking about this is that in previous users who were using alcohol during adolescence but are now abstinent, these alterations continue even after periods of abstinence. So, and here's one of the most striking facts to me, and what has made me really interested in this field actually, is that early adolescent drinking onset is associated with a 4 to 5-fold increase of developing an alcohol use disorder within the lifetime. So this is really increasing the odds. The earlier that you start drinking, especially binge drinking, can really lead to a lifetime, sometime within the lifetime, developing an alcohol use disorder. So, however, this is a lot of things that we're talking about with humans, and alcohol is not always used in isolation and often is not used in isolation, and can be compounded by other comorbid drug use. You know, some people drink when they smoke or things like that, so there can be nicotine and alcohol on board. And so in order to kind of really pinpoint what are the effects of alcohol drinking in adolescence, we really look to animal models in order to be able to look at this and see what are the exact effects of alcohol without the potential of these other comorbid drug use occurring as well. And so what's really great is that adolescence is a period that's conserved across many mammalian species. So brain maturation during adolescence is really well conserved across mammals, and we've shown that previously. So things that we mean in this brain maturation would be neural pruning is conserved. We see this gray matter volume reduction as adolescence continues and white matter volume increase, and this is well conserved across species. We see this peak activity in the mesolimbic dopamine system, right? This is the reward system. This is very active during adolescence, and we see this across species. There's also less inhibition on the prefrontal cortex compared to when in adulthood. So, right, you have this peak in your reward-seeking kind of circuitry and this inhibition of the prefrontal cortex, and we see this conserved across species. And the hypersensitivity to the receipt of reward, this is something where you're just activated more and you're more sensitive to getting that reward, the pleasurable effects of that reward. And there's this really critical aspect of social context, and this is shown throughout our species, right? Especially I use more of a rodent, which we'll get into, and social play and social interaction is hugely important across mammalian species. So how do we then bring this into rodents? So we use rats in our lab. How do we model this in binge drinking? We know that adolescence is conserved in rats very similarly as into humans, so we know that this is a good model for us to use, but now how do we do it, right? How do we make them drink? And so the thing is that the adolescence in rats is much quicker than in humans, obviously. So PMD stands for postnatal day, and so adolescence spans from approximately postnatal day 28 to 55 in the rat. And so this is a little bit conservative. They can go a little bit further or be a little bit earlier, but this is, you know, the main range of adolescence in the rat. And we use various methods of administration of ethanol. So adolescent rats are actually a little bit smarter than adolescent humans. They don't tend to just self-administer alcohol readily or to the binge levels that human adolescents will use. And so we use various models of administration to kind of experimentally administered alcohol, right? So you can do intragastric, which is to actually place the ethanol into the stomach, and that is what we use. There's also some inhalation, or you can use an intraperitoneal injection, or there's other oral ways that you can kind of make them, like force them to drink it. But the intragastric is where you place it directly into the stomach, and that is what we use. We found this to be a really good model because it allows the ethanol to be absorbed into the system similarly as it would be into a human adolescence, through the gastric system. We use an intermittent schedule, right? So they're not getting alcohol every day. And this is to binge those same periods in human adolescence where you see the high levels of use on one day, but then they go through periods of abstinence, right? So to do that, we do a 2 days on, 1 to 2 days off schedule, which I'll get into a little bit more. And so binge levels are at greater than 80 milligrams per deciliter of blood plasma. That would mean that they hit a binge level. That's by the National Institute of Alcohol and Alcohol Abuse. And so in our model, we tend to use intragastric gavage, where we give 5 milligrams per kilogram of ethanol. This really gets us a reliable to approximately 200 milligrams per deciliter. So they are really going a lot higher. And this is really looking at that heavy binge drinking in these animals. And we give them 10 doses over 16 days. And so that models that 2 days on, either 1 to 2 days off. And we dose them from PND day 30 to 46, so right kind of in the middle of this adolescent span in these animals. And so we know that by using these methods, we get similar alterations in the hippocampus in adulthood that we find in the human population. So within the hippocampus, we see learning and memory deficits, as I had said, were previously shown in the adolescent human population. We also see volume decreases in the hippocampus and other areas in both clinical and preclinical work. Alteration in hippocampal inputs. There are changes in morphology in the hippocampus, and there's increased immune signaling. So we know that this is a pretty good model for adolescent binge drinking in our rodent population. And so we and others that we collaborate with and other labs as well have found that AIE, so again, this is that adolescent intermittent ethanol modeling in the rodent population, so I'll be calling it AIE. So AIE reduces cholinergic innervation into the hippocampus. So within the basal forebrain, which is called CH1 and 2, these guys send the cholinergic input into the hippocampus, and they are the primary innervator of cholinergic input into the hippocampus. This is really important for facilitating memory-related neuronal function. Again, the hippocampus, and we focus a lot on the dorsal hippocampus, which is important for spatial memory and function. And so this projection of cholinergic innervation is really, really integral for this memory-related neuronal function. You can look at choline acetyltransferase, also called CAT, and this is a marker for cholinergic neurons, not just interneurons, sorry. And what we have found is that in the in the CH1 and 2, this is the area that projects to the dorsal hippocampus, the CH1 and 2. Animals that receive either water, which are labeled here as controls, they are not getting ethanol in adolescence, or AIE, Adolescent Intermittent Ethanol, that CAT immunoreactivity is reduced in CH1 and 2, and this is really indicating that there is reduced innervation into the hippocampus, cholinergic innervation to be specific. And so it creates this chronic deprivation of this cholinergic input into the hippocampal circuits. And actually one thing that a collaborator of ours has shown recently but has not published yet is that these neurons in this region, so in CH1 and 2, they're actually not reduced. So this reduction in CAT marker and immunoreactivity in the CH1 and 2 is not actually indicative of a reduction of neurons. So it's the same number of neurons, they just seem to be changed into a different type of neurons. They're no longer expressing CAT, but they may be expressing other neurotransmitters and things like that. So this is really interesting and something that we're moving forward with. It's not a complete reduction of this region, but it's just a reduction in CAT. And so we have also found that in the hippocampus, and now we're moving from CH1 and 2, which provides that cholinergic input into the hippocampus, now looking at the hippocampus, we have found reduced neurons in the hippocampus and an increased immune response in CA1 or the dentate gyrus, in CA1. So we found reduced neural numbers in the hippocampus following AIE. So this is, again, I don't think I made this completely specific, but we are looking in adulthood. So adulthood starts at postnatal day 70 or greater. And so here we have given these animals AIE in adolescence from PND 30 to 46, but now we have let them age up into adulthood. And here we're seeing this reduction in neural numbers in the hippocampus. These are, persistent changes that continue. And we've also looked at some immune response, such as astrocytes. So these guys are really integral for synaptic formation, connectivity, and really the function of the hippocampus as it goes with working with in the tripartite synapse. And so we have found that astrocytes are actually more reactive. And so reactive astrocytes are larger. And so after AIE, we see these larger astrocytes, as you can see in this bottom right corner here, follow in adulthood. So this is kind of a persistent change that we see that's showing a reduction in the number of neurons and this kind of increased immunoinflammatory response and a pattern of immune response in the hippocampus following this history of AIE, so looking in adulthood. So we've also found that there is a loss of adult neuroimmune activation and then the loss of neurogenesis. So neurogenesis is new neurons being formed, right? And we look at that through the marker double cortin. So we're looking at double cortin immunoreactivity here. Our control animals who received water in adolescence are in the open bars and the AIE animals are in the closed bars. So if we look here on the left panel, looking at the no exercise group, that's closest to the y-axis, we can see that with no exercise, so kind of a standard model of AIE, we see this reduction in double cortin immunoreactivity after AIE in adulthood. But what's really exciting about this is that if you look at the exercise group, so this was a model of prevention, we see that the AIE animals look just like those water controls, and we actually can reverse these changes in the hippocampus and this reduction in neurogenesis, which is really exciting because it shows that even though these changes are very persistent and they continue into adulthood, they are reversible, right? And so what we're also looking at here is cleaved caspase immunoreactivity, and what we see is that, so this is a death receptor, this is a marker of cell death, right? And so what we see is that this marker of cell death is increased in AIE animals in these darker bars compared to the control animals. However, exercise reduces this elevation in the AIE animals. So again, it is reversed by exercise. And another type of cell immunoreactivity, we're looking here at NF-kappa B. And so what we see here is that on the left panel in the no exercise group, the AIE animals have elevated NF-kappa B in adulthood immunoreactivity, and that this can again be reversed by exercise. So we see these alterations that are caused by AIE that persist into adulthood, but again, they are able to be reversed by exercise, so they are reversible. And so from this, where we see this reduced cholinergic innervation, increase in inflammatory response, or reduced cell population, and increased cell death and reduced neurogenesis in the dentate gyrus, we wanted to again look at these in our model. And we also had some other information, sorry, forgot about this slide here, where we're going to talk about the neuroimmune changes that occurred in the prefrontal cortex that also implicates some more of these neuroimmune responses. So in the human, we see RAGE, which is a receptor for DAMPs, danger-associated molecular proteins pathways. And so this will elevate kind of our cell death machinery as well. And HMGB1 is an activator of RAGE, TL4. So we see these all increase in the human alcoholic in the prefrontal cortex. We've also seen it in our adolescent rats. And so this brought in our want to kind of look at these markers in the hippocampus as well, right? So we have our double cortin, our neurogenesis that is reduced after AIE in the hippocampus that we know, as well as some cell death markers. And then additionally, this could be looking at more of a mechanism if we look at RAGE and HMGB1, which can be these activators of these cell death machinery. So what we wanted to know is that could we replicate this in the hippocampal neuroimmune and neurogenesis deficits, and can we reverse these effects? And we wanted to reverse these effects pharmacologically, not with exercise. And so we looked to Donepezil, which is also known as Aricept. This is a selective reversible cholinesterase inhibitor. So we really wanted to target this kind of reduced innervation into the hippocampus and see if this is what's kind of causing these learning and memory deficits. So this is used clinically to combat memory-related cognitive deficits, especially specifically working memory deficits, and this has been used in several animal models, as well as obviously it's FDA approved and clinically used. So adult rats with the history of AID have that reduced cholinergic innervation into the hippocampus. We thought that we could possibly reverse this using donepezil. Okay, so for our study design, we used intragastric gavage. Our animals arrived on PND 25 and then they habituated to the colony room until PND 30 when they began their AIE treatment. Again, this is 2 days on, 1 day off, 2 days on, 2 days off for 16 days. So it gives 10 doses over 16 days from PND 30 to 46. They aged up into adulthood, and then they received 2.5 milligrams per kilogram of, again, intragastric gavage of donepezil for 4 days, and they were sacrificed 2 hours after their last dose. So the targets that we looked at were targets of neurogenesis, cell death machinery, neuroimmune signaling, epigenetic markers, and again, we were looking for this reversal by donepezil in these studies that I will explain. So first off, we wanted to replicate that DCX, so that marker of neurogenesis that had been found previously. And so what we found was that AIE exposure reduces in the dentate gyrus neurogenesis, and this was reversed by donepezil. So if we look over here at our graph, we see that we have DCX immunoreactivity. In our open bars, we have animals that were treated with vehicle, which was water. And in the black bars, we have the animals that are treated with donepezil. We have AIW in the first grouping on the x-axis and AIE in the second grouping. And so what you can see is that animals that were treated with vehicle and had a history of AIE had reduced DCX immunoreactivity compared to their water controls, but donepezil reversed this deficit and was able to make these animals look similar to their water-treated controls. All right, and over here on the right, we just have the representation of the DCX immunoreactivity. And so this is a marker of neurogenesis, and we're seeing that there's reduced neurogenesis in the dentate gyrus after AIE, but this can be reversed by donepezil. So this is a reduction of new neurons that is reversible. We then wanted to look at some cell death machinery in the dentate gyrus as well and see if this was reversed by donepezil. So we looked at active caspase-3 immunoreactivity, and we again saw that in our AIE-treated animal, AIE-treated vehicle animals, there was this elevation of activated caspase-3 immunoreactivity that was then reversed by treatment with donepezil in adulthood, right? So this is so important because our treatment is in adulthood after these persistent changes have occurred and lasted for about 25 days, but then treatment reverses these alterations. And so on the right-hand side here, you can see our representative images. And then we also looked at death receptor 3. This is another cell death machinery marker. And so what we see is that this is very elevated in our AIE-treated vehicle animals and that this was reversed by treatment with donepezil. So both of these cell death markers can be, they are elevated after AIE in adulthood and they are reversible by donepezil. So we also wanted to look at some neuroimmune function and start to get at the mechanism behind how this is happening. So we started to look at epigenetic plasticity again, and here we are in the dentate gyrus. So first off, we looked at NF-kappa B. We again found that this was elevated after AIE and reversed by donepezil. So NF-kappa B is really important for cytokine production and immune response regulation. So this elevation is showing this elevation of neuroimmune function after AIE, but then again, reversal by donepezil. We looked at pro-inflammatory gene activation. So this is the RAGE receptor. This is elevated after AIE and then again reversed by donepezil. And HMGB1 is one of the activators of the RAGE receptor. And this is the only one that we can, because I probably am starting to sound like a kind of broken record here, reversed by donepezil, but this was not reversed by donepezil. So this is a danger-associated molecular pathway agent, and so as we see here, it is not affected by AIW or water treatment with donepezil, but in our AIE-treated animals, it is elevated, and this is not reversed by Donepezil. So, and HMGB1 has some really interesting signaling and methods on which it goes. So this is actually really interesting that it's not been reversed by Donepezil and cause for some future investigation. But lastly, we looked at an epigenetic marker. So this is kind of an indicative of repressed transcription. And what we found is that after AIE in adulthood, if they were treated with water, we find this elevation of H3K9 methylated 2 immunoreactivity, and that this again was reduced by Donepezil and no different than the water-treated control animals. So this is kind of starting to paint this picture for us of potential mechanisms by which we find this reduction of neurogenesis and elevation of cell death machinery within the hippocampus, and is kind of giving us a little bit of a hint that we should be investigating, these elevation and rage receptors and its associated activator HMGB1 as well as NF-kappa B, which can cause, this elevation of immune responses and has a lot of downstream signaling. And this epigenetic marker showing repressed transcription kind of gives us this really great new area that we should be kind of heading into. So this is the potential mechanisms that we're going to be investigating behind this reduced neurogenesis and increased cell death. And so just in summary, we went through today kind of some seminal findings that we had in the hippocampus and the dentate gyrus, where we showed that AIE reduced neurogenesis, and this was reversed by Donepezil. Additionally, we found that AIE exposure increased cell death machinery in the dentate gyrus, and this was reversed by Donepezil. Then we started to look into the mechanism of how this neurogenesis and cell death machinery alterations are caused by AIE, and we kind of looked into neuroimmune function and epigenetic plasticity in the dentate gyrus. We found that for the most part, Donepezil reversed those alterations except for HMGB1. And so this is leading us into some future directions where we're going to continue to kind of look at the mechanism behind the neurogenesis deficits and increase neuroimmune signaling of AIE. We want to kind of continue to figure out what is the direct mechanism behind all of this. And we also want to take these findings and kind of look into the prefrontal cortex and see if we see these alterations and if they are reversed by Donepezil. Our lab has recently been taking kind of a more astrocytic component as well, so we do want to look in both the hippocampus and the prefrontal cortex to look at the astrocytic involvement, which is what we've been doing most recently. So with that, I just want to say thank you to my lab. I am, again, a postdoc in the Swartzwelder lab, so I work with Scott Swartzwelder and, again, our awesome technicians and undergraduates, Sandra Kibble, Kira DeBester, Sierra Hodges, Amelia Bell, George Kramer, as well as our collaborators, Fulton Cruz, Wen Liu, and Sean Atchison, who helped get this project. And of course, our funding, we are a part of the Neurobiology of Adolescent Drinking in Adulthood Consortium, the NADIA Consortium, who primarily funded this work through NIAAA and these funding sources. So thank you for coming to my talk.